magnesium chloride and ethylene glycol tetraacetic acid are added to the buffer and calcium is left out. This buffer chelates calcium which blocks classical pathway activation. Rabbit red cells are used as the indicator because this provides an ideal surface for alternative pathway activation. An additional means of testing for alternative pathway function is via ELISA. One such test can detect C3b-BBP or C3BP complex in very small quantities. Microtiter wells are typically coated with a bacterial polysaccharide to trigger activation of the alternative pathway. One test has been developed that can determine the activity of all three pathways: the classical pathway, the alternative pathway, and the mannose binding lectin pathway. In this test system, strips used for the classical pathway are coated with IgM. Strips for the alternative pathway are coated with the lipopolysaccharide, and strips for the mannose binding lectin pathway are coated with mannose. Such testing is easy to perform and is not dependent upon the use of animal erythrocytes, which may be hard to obtain. Deficiency can be detected using combined test results. Now we're going to talk about the interpretation of laboratory findings. So, the decreased level of complement components or activity may be due to any of the following: decreased production, consumption, or in vitro consumption. The third condition must be ruled out before either of the other two is considered. Specimen handling is extremely important. Blood should be collected in a cloth tube with no serum separator. The tube should be spun down and the serum should be frozen or placed on dry ice if it is not tested within 1 to 2 hours. If a specimen has been inadequately refrigerated, been subjected to multiple freeze thaws, or been in prolonged storage, the results may be invalid and the test needs to be repeated with a fresh specimen. Control serum should also be included with each batch of test sera. A typical screening test for complement abnormalities usually includes determination of the following: C3, C4, and factor B cells levels, as well as hemolytic content. Testing for product of complement activation such as C3A, C4A, C5A, and BA, as well as breakdown products including C3DG, IC3B, and C4D can also be performed as a means of monitoring inflammatory process such as rheumatoid arthritis and systemic lupus erythematosus. Table 6 to 5 presents some of the possible screening results from ELISA testing and correlates this with deficiency of individual factors. An understanding of this pattern may be helpful in differentiating hereditary deficiency from activational states that consume available complement components. Additional testing would be necessary, however, to actually pinpoint hereditary deficiencies. Now we're going to talk about the complement fixation testing. The complement itself can actually be used as a reagent in this test known as complement fixation because complement fixation occurs after the binding of antigen and antibody. Uptake of complement can be used as an indicator of the presence of either specific antigen or antibody. This technique has been used in the detection of viral, fungal, and rickettsial antibodies. The test involves a two-stage process: a test system with antigen and antibody, one of which is unknown, and an indicator system consisting of sheep red blood cells coated with hemolysin, which will lies in the presence of complement. To destroy any complement present, patient serum must be heated to 56 degrees Celsius for 30 minutes prior to testing. Then dilution of the serum are combined with known antigen and a measured amounts of guinea pig complement. Guinea pig complement can be used for testing because complement is not species specific. If patient antibody is present, it will combine with the reagent antigen and complement will be bound. The test can also be performed with reagent antibody to detect the presence of antigen in the sample. The second steps involve adding sheep red blood cells that are coated with hemolysin. After an additional incubation, the tubes are centrifuged and then read for hemolysis. If hemolysis is present, this means that no patient antibody was present and the test is negative. Lack of hemolysis is a positive test. Results are expressed as the highest dilution showing no hemolysis. The use of control is extremely important to ensure the accuracy of test results. This includes running known positive and negative sera, an antigen control, a patient serum control, a cell control, and complement control. The antigen and patient serum control test for the ability of complement to bind in the absence of antibody. This control shows lysis with the sheep red blood cells. The cell control is a check on spontaneous lysis. Lysis should be absent or extremely low. The complement control tube should show lysis to indicate that the complement is working correctly. Due to the amount of manipulation required and the need for standardization and numerous controls, the use of complement fixation in the clinical laboratory is limited. Now, to summarize our discussion, the complement system is a series of more than 30 proteins normally found in serum that play a major role in phagocytosis and clearance of foreign antigens from the body. While the end product of complement activation is lysis, other important events such as opsonization, increase in vascular permeability, and chemotaxis of monocytes and neutrophils take place along the way to enhance host defense mechanism. Most of the proteins of the complement system are inactive enzyme precursors or zymogens that are converted to active enzymes in a very precise order. One such means of activation in the classical pathway is which is triggered by antigen-antibody combination and is involved 9 of the proteins. The alternative pathway acts as a natural means of defense against infection. It is triggered not by antigen-antibody combination but by the presence of microorganisms. The lectin pathway also involves non-specific recognition of microorganisms by means of sugars such as mannose that are common constituent of microbial cells walls, but that are distinct from the found on human cell surface. Although initial activation of this pathway involves proteins that are unique to each. C3 serves as a major role in linking all three pathways. Cleavage of C3 to C3a and C3b represents one of the most significant steps in all pathway. Because C3b is a powerful opsonine that greatly enhances phagocytosis. C3a is an anaphylactoxin that causes release of reactants from mast cells that amplifies the inflammatory response. The end result is lysis of cell due to production of a membrane attack complex that drills holes in the cell membrane. If activation of the complement system was to proceed uncontrolled, tissue damage and life-threatening systemic effects might result. Therefore, several proteins act as regulators to keep the reaction localized and ensure that infectious agent and not self-antigen are destroyed. Some of these are free-floating in the plasma, while others are found on cell surface such as specific receptors. One class of membrane receptors on host cells act in conjunction with complement components to amplify the immune response. These receptors bind antigen-antibody complexes and facilitates phagocytosis. Other functions may include enhancing activity of phagocytes and production of increased memory B cells. Complement plays a major role in several disease states. Decreased complement levels or a decline in electric activity can indicate either hereditary deficiency or extreme activation of the system. Paroxysmal nocturnal hemoglobinuria and hereditary angioedema are two conditions that result from missing or very low production of certain regulators. Deficiency of some components, especially that of C3, may cause an increased risk of life-threatening infection or glomerulonephritis due to a lack of clearance of immune complexes. Several laboratories assays have been devised to detect abnormal complement levels, one of which, the CH50 hemolytic assay, uses 50% lysis of standard concentration of antibody-sensitized sheep erythrocytes as an endpoint. This measures the presence of all protein involved in the classical pathway. A similar assay, the AH50 assay, measures the functioning of the alternative pathway. Other assay using RID or ELISA's techniques measures level of individual components. Proper handling of specimen is essential to ensure correct interpretation of laboratory testing. Complement fixation tests use complement as a reagent to detect the presence of antigen or antibody. Antigen and antibody are allowed to combine with a measured amount of complement. Then indicator-shaped red blood cells are added. They are coated with hemolysin anti-sheep cell antibody, and if complemented has not been tied up in the first reaction, then lysis of the cell occurs. Thus, lysis is a negative test indicating that either antigen or antibody was not present. As in all complement testing, the use of standard and controlled is necessary for accurate results.